Hello everyone, I'm Koyan and today we are going to learn how to answer questions related to Form 4 Chapter 2 Cell Biology and Organization. So for Form 4 Chapter 2 to 7 are the fundamental chapters you must at least know the basics of it because it surely will come out in a structured question. So now let's take a look at this question. Diagram 2 shows organism R that lives in a fresh water. So state the kingdom for organism R. Okay, so students might having a panic attack at this first question. What is actually a kingdom? Can you recall what is that? It's actually from Form 5 Chapter 8, where you learn a lot of classification. I know students probably dislike that chapter. So let me show you this classification right here. So there are actually domain, kingdom. Yeah. So there are actually domain, kingdom, phylum, class, order, family, genus, and species. How do you actually remember? I actually remember it through this way. Does king prefer candy over fresh green salad? Yeah, that's how I actually remember. So for kingdom, they actually branch out into six of them, which are Archaebacteria, Eubacteria, Animalia, Plantae, Protista, and fungi. So this one, can you actually guess what is it? It's actually a Protista. Okay. So since it's a protozoa, so it belongs to a Protista kingdom. So for the kingdom, you have to write it as Protista. Maybe a lot of students may lose mark on this part lah because um this really requires memorization where you have to know the classification and how many types it branches out, and this one belongs to which kingdom. And for Protista, right, for your information, it actually branched out into other three of them. So Protista, there are actually animal-like one, or plant-like, or another one which is quite special, which is slime mold. But the one animal-like is the protozoa where we are learning. And then the plant-like is actually the algae. Yeah, it branch out to another three of them. So now let's move on to see label oral groove and cilium in diagram 2. So how do you actually label it? So first of all, you have to at least know which is which lah. Okay, so this one is basically the hair-like structure one is basically a cilia. I'm just labeling it to you, although the question didn't ask about it. And another thing, the flower one is actually a contractile vacuole. For paramecium species is special lah. The contractile vacuole is in a flower shape. And moving on, let's take a look at this one. They said label overgrowth and cilium. So yeah, so sorry for that. You better write it in cilium. And then where is the oral groove? So oral groove is where the food will be transferred into. So can you see the opening of this paramecium species? So yeah, this is basically is around here. So this one is the oral groove. So how do you label it? For biological drawing, it is better to use a straight line. So you have to use a ruler and then you just point it out right here. Okay, and you write it out oral groove. Okay. Meanwhile, for the others, this one is basically, I mean, the question didn't ask you, but I'm going to label it to you in case you don't know what are these structure. So for this very darkened shade one, is actually a nucleus. And then, yeah, the jelly-like substance right here is all the cytoplasm. Meanwhile, what about this circle and then containing like granules inside? These are actually food vacuole. food vacuole. And then this is the organelle. So yeah, that's how you actually label them. But yeah, the question actually required you to label these two only. So now let's move on to the next question. Explain how structure K helps organism R to live in the freshwater environment. So what is structure K? Structure K is a contractile vacuole. Since they live in a freshwater environment, they actually need this contractile vacuole to undergo osmoregulation to regulate the water content. So how does it help the organism are to live in a freshwater environment? Because the freshwater, they contain like a lot of water, high water potential, so they need to regulate it. So they need this contractile vacuole, which is structure K. So the main process they are in charge of is osmoregulation. So the main point you have to write is osmoregulation. Osmo means water, so regulate the water lah. So it's called osmoregulation. And then you must state this osmoregulation as the main point. Then you only elaborate. So how does it help for this? So you basically have to write that when the water diffuse into K via osmosis, or you can write by osmosis, the K, which is the contractile vacuole, will expand until it reaches a maximum size. So when it reaches the maximum size, can you imagine what happened? Is it going to burst? No, it's not going to burst. It will actually contract to expel the excess water. So K will contract to expel the excess water. So the keywords you have to write is water. The movement of water is via osmosis. And then you have to write this word called diffuse. It's better lah. Don't write move into. It's better, more ideal to write diffuse via osmosis. And then it will continue. The contractile vacuole will expand until when it reaches the maximum size, they will eventually contract to release out the excess water. So it will contract to expel the excess water. So yeah, that's how it helps organisms out to live in a freshwater environment. Basically, they are asking you to explain the role of contractile vacuole. Alright, now let's move on to the next question right here. El Nino phenomenon has threatened the habitat of organism R. Explain how organism R reproduces in that environment. So what is El Nino phenomenon? I think you probably still can recall a bit during our geography class about El Nino, La Nina phenomenon. But basically, what are they trying to hint you? They are actually hinting you that explain how organisms they reproduce in that environment. So for reproduction of paramecium species, right, in an ideal environment, they will actually use binary fission to reproduce. However, in an unfavorable condition, so this one is during a favorable condition. But during an unfavorable condition, they will use another way to reproduce because they want to reproduce, so they have an alternative to it. So El Nino phenomenon is where it has extreme weather that threaten the habitat. So the habitat now is not ideal. So not ideal means it's unfavorable condition. They are hinting you about this unfavorable condition. So how does organism are reproduced? It's not through binary fission since the condition is not favorable anymore. So during an unfavorable condition, they will actually reproduce through a sexual reproduction, which is called conjugation. So the maxim actually accept if you write sexual reproduction, but if you can recall the specific sexual reproduction, you can go on and write the conjugation instead. Because sometimes biology they really need your answer to be specific. But if you really can't think of that specific answer, then you have no choice. You can write sexual reproduction. Meanwhile, I want to recall about the amoeba species. For amoeba species, during a favorable condition, right, they also reproduce through a binary fission. But for unfavorable species, for amoeba species, okay, this one is also the same. But for unfavorable species, they will actually reproduce through spore formation. That's something you have to take note of. They are actually different when it comes to unfavorable condition for these two paramecium and amoeba species. This one is conjugation. Yeah. All right. So now let's continue to move on to the next topic. This is also talking about the paramecium species. So it's very direct. You can see a shoe sole right here, shoe sole shape, and a hair-like structure, which is the cilium, and Q, which is the oval groove where they transfer the food particle. And then, yeah, that's all. Sorry for that. I think it's better to label it like that, this way. Okay. All right. So let's take a look. What is the structure P and Q? So it's very obviously known that this is a flower-like structure, very specific, very special. So it's a contractile vacuole. And then Q is where they transfer this food particle. So it's a oval groove. Alright, now let's take a look. Explain how food particle is able to enter structure Q and being digested. So how do they actually transfer the food particle? So for human, we probably just use our hand and then eat the food like that. I mean, we use utensil to be more polite. But yeah, basically we use our hand. But what about this paramecium species? They don't have hand, but you can see they got this hair-like structure, right, surrounding them. And in detail, you can see that they are the one that help out to transfer the food particles inside. So you have to emphasize the role of the cilium. So now let's answer this question. Yeah. Cilium, they will actually have this rhythmic beating when they are doing movement. When they move from one place to another, they will use rhythmic cilia beats. And other than that, other than movement, cilia role is to transfer the food particle. So this cilium beats, it will transfer, or you can write it as push the food particle into where? Food particle into our groove. Or you can write structure Q if you are not confident. So structure Q, or you can write it as oval groove. And how do you continue to explain when it comes to digestion of it? So the food vacuole. So for us human, we have the digestive enzyme to digest it, right? Same goes to this paramecium species. But first of all, they have to undergo this process where food vacuole will combine with lysosome. And then this lysosome, they have this lysozyme enzyme. So the food particle is hydrolyzed. Hydrolyzed means it's broken down with water by lysozyme. A lot of students will get confused between lysosome and lysozyme. So this zyme, the one ends with the zyme, is the digestive enzyme. Lysosome is the organelle we learn, okay, that will actually break down worn out organelles for us animal cell. But for paramecium species, they are the one that helps up with the digestion. Now let's move on to question C. Predict what will happen if organism is placed in marine water. So it's more of a prediction K-BAT question. It's not a very direct question for you. So what is the condition of marine water? Marine water, because in a normal ideal situation, paramecium species actually live in a fresh water. But now they place it inside this marine water. Marine water, have you ever went to the sea and then you swim, then you accidentally swallow some seawater? Yeah, it's extremely salty, right? So it means it's a high concentration salt water lah. So this salt water, if you think of it in a biological way, so how do you explain the condition of the salt water, which is the marine water? Marine water, they have a high salt concentration, right? So when it has a high salt concentration, means it has a high solute potential. So when it has a high solute potential, it is hypertonic to the paramecium species. So you have to mention what is the condition of the marine water. And then when the paramecium is placed in this marine water, what will happen for the water? And wait, let me write it down. Water is hypertonic to cytoplasm of paramecium species. And you must write paramecium species to be safe lah. Together, not just paramecium. So now let's imagine this situation. This is the paramecium species. I know it looks like a bacteria, so sorry for that. Okay, so this is a paramecium species. And then now you are placed in the marine water. So here got a high salt concentration means a high solute potential. When it has a high solute potential, means that it has a low water potential right here. Meanwhile, for your paramecium species, they have a higher water potential. So you know the movement of water is through high water potential to low water potential. So it will eventually diffuse out via osmosis. So you have to explain what happened next, whereby the water molecule diffuse out from cytoplasm from where to where of paramecium species via osmosis. So when you are talking about the water movement, right, it is very important for you to write diffuse in or diffuse out from where, and then the process, osmosis. So these two are the keywords. And then what happens next? When the water keep diffusing out, can you actually imagine what happened to the situation? You get a lesser water, and then when you live in a condition when the paramecium species have a very low water content, it will eventually lead to dehydration. So yeah, it will actually die lah. So paramecium species undergoes dehydration because the water keep losing out from its cytoplasm. Dehydration due to lack of water. Because during this time, the condition is too extreme that the contractile vacuole can't really help out to retain the water. But the contractile vacuole, they will actually stop contracting to prevent water from losing out. So they will not contract so that water we won't lose. But at the same time, the water from their cytoplasm will actually lose out. So eventually, in the end, in the long term, yeah, it eventually dehydrated lah. Now let's move on to the next question. So this one is basically animal cell. And this animal cell, you can see all the organelles right here you have learned before. It's very obviously known that Z is a mitochondria. Chondrion. This one is singular. And then, can you actually identify what is Y, what is X, what is W? These three are actually students get confused easily. So let's take a look at this part. Supposedly the diagram, to be precise, it's better for the rough endoplasmic reticulum to attach to the nucleus. But this diagram, yeah, no comment. So for this, how do you actually identify rough endoplasmic reticulum? Since it's rough, right, means it has ribosomes that attach to its surface. So it is rough since they have ribosomes. So this one is basically the rough ER. I just write in short form, but please do not write in ER form. Okay. And what about smooth ER? Can you actually differentiate the left one or the right one is the smooth ER, or is it a Golgi apparatus? So this Golgi apparatus, right, their membrane sac is actually not continuous. You can see in this form, they are actually not continuous. They are not joining together. Meanwhile, for the smooth ER, you can see that they are joining together like that. Yeah, they are actually joined together. So this is actually the smooth ER. Another way to identify is that you can see this circle part, right? It's not air bubble. It's actually the secretory vesicle and the transport vesicle. So this secretory vesicle, it will butt off from the Golgi apparatus lah, because this Golgi apparatus, it will modify and then package, then the protein, let's say extracellular enzyme, it will be secreted out through secretory vesicle and then fused with the plasma membrane to be transported out. So it surely has this vesicle right here. So this is another way to identify whether it is a Golgi apparatus. So this one is Golgi apparatus. So this is, yeah. Be in mind that, remember, the spelling is really important, and the Golgi, the G is in a big capital letter. Then let's move on to this one. Match the cell component W to its function. What is the function of Golgi apparatus? So it actually, okay, synthesize protein. Who actually synthesize protein? It's actually the ribosome that synthesize protein. Who is the one that hydrolyze protein? Hydrolyzed protein is actually, probably enzymes are the one that hydrolyze protein, like protease enzyme. So yeah, it's not a function of Golgi apparatus. Golgi apparatus, they play a role in modifying the protein. For example, modify protein into the enzyme, because enzyme is made up of protein. So W, this Golgi apparatus, the function is modify protein. Now let's move on to the next question right here. The membrane of cell component X and Y extend from the nuclear membrane. State one difference between cell component X and Y. So what are the difference between them? So the most obvious difference is the ribosome, right? So you can write cell component X. Wait. So X is the smooth ER. Meanwhile, for the Y is the rough ER. So smooth one means it doesn't have a ribosome attached to its surface. Ribosomes attached to its surface. When you're doing differentiate, right, you have to write the format which is while, or you can write whereas, cell component Y, blah, blah, blah. Cell component Y has ribosome attached to its surface. What about other thing you can actually compare? You can actually think of their, other than structural difference, you can think of their function. So cell component X smooth ER, they actually synthesize or transport lipids. So synthesize lipids, or you can write glycerol. So and followed while cell component. Okay, I write in a very cursive manner. So this one is actually the one that transport protein synthesized by ribosome. So you can just write transport protein will do. If you want to be more detailed, you can write synthesized by ribosome. And this one only 1 mark, so you can just write out one differences will do, because I'm trying to give you the most complete answer. But you just write out one, then you just get a full mark right there. And give the reason why the density of cell component Z is abundant in liver cell. So cell component Z from the diagram is actually the mitochondrion. So mitochondrion, you think of the reason. What is the function of mitochondrion? Is actually to generate energy, right? The function of the mitochondrion is to generate ATP energy. So how are you going to explain? When you write generate energy, it actually doesn't really answer well to the question, because they are talking about abundance. Because in our whole body cell, there are a lot of parts which have the mitochondrion, but the distribution of the mitochondrion is different. For the liver cell, it tends to be more. So you have to emphasize it is to generate more energy. You have to write out the more energy. So to generate more energy in form of ATP, if you want to be precise. So why do you need this energy in this liver cell? I know this is only 1 mark, but if you want to know in detail, it's actually for the function of the liver. So what does the liver do? Because there are actually a lot of metabolic processes that require energy. So for liver, is for they need energy for the process of, so liver actually does a lot of things like assimilation, or maybe even deamination, how it metabolizes the amino acid, and maybe detoxification, detoxification. So if you drink alcoholic drinks, yeah, you are giving your liver a hard time to detoxify it. Okay, so yeah, this is actually more in detail for what process lah. And this you actually get 1 mark. and this one is another mark if you explain it more. But yeah, the full mark is just 1 mark lah. And now let's take a look at question C. The cell has been infected with a bacteria. Explain the cell component that can destroy the bacteria. So which cell component can actually overcome this bacterial infection? Is actually the one that have the digestive enzyme that can eat the bacteria, that can break down it. So who's this hero, this organelle? Is actually the lysosome, like what you learned just now in the paramecium species. That one, they have a digestive enzyme for their nutrition. They have to break down the food. But this one is talking about animal cell. So when it infected with bacteria, yeah, the lysosome that contains lysozyme digest, or you can write break down bacteria. Because one of the function of lysosome is to break down worn, damaged organelle. So the another function is, since it has this lysozyme, so yeah, it can help out to break down the bacteria. Alright, now let's move on to this one. Is chapter 3 already. So yeah. So for this chapter, yeah, basically you have to focus on the paramecium species. And yeah, that's all for this lesson, and I'll see you in the next video. Thank you so much for your time. Good luck on studying.